Hello students, welcome to the 2021 Ohio State test, and this is part 3. So let's get started. In 1961, scientists used repeating RNA segments such as UG to build amino acid chains. Based on the amino acid chains built, they assembled the codon chart shown. And there's our codon chart. So let's look at question number 11. The following question has two parts. First, answer part A, then answer part B. Which diagram shows how RNA is read to produce an amino acid chain? So remember, DNA to RNA. DNA makes messenger RNA inside the nucleus. That's called transcription. And then the messenger RNA leaves the nucleus, goes outside to the ribosomes, and starts making protein by putting the amino acids together. But the DNA gives the messenger RNA a code, and that code is called a codon. And this is an easy answer to discover. First of all, it's C, right? The codon is always 3 bases that the DNA gives to the messenger RNA. And so there's 3 bases, that's our codon. So that's a real easy answer to get. Now let's go to part B. Which amino acid chain would be built from the RNA segment UG? Well, let's look here. UGU, so let's look on our codon chart. UGU is CYS, right? Okay, now let's see what the next one is. That would be GUG. So we go down here, G, U, and G. And that would be Val. Ooh. So we got UGU, GUG, and we got UGU again. So we got UGU, and that's the cis. So our answer has to be C. That's pretty easy. Question number 12. A scientist investigates the effects of a single base substitution mutation in a 3-base DNA sequence. This 3-base DNA sequence codes for an amino acid. A table containing some of the scientist's data is shown. Fill in the original strand boxes in the table based on the amino acid produced. That's A. B. Fill in the mutated strand boxes in the table based on the DNA sequence. So let's look and see what we have here. We have the original strand of DNA not available, the messenger RNA not available. But they did give us an amino acid produced. So by looking at the amino acid produced, we can discover the messenger RNA. So let's look here. Let's find the amino acid. There it is, TRP. So let's use the codon chart to find out the messenger RNA. That would be UGG. How about that? So now that we know UGG, we know our original DNA before the mutation occurred. It would be ACC. There you go. And now here's our mutated strand. See, the A's changed to a T. Okay, so this is our mutated DNA. So we know trees, we have an apple, and we have a car, we have a garage. And now to find our amino acid, all we have to do is look up A, G, and G, and that is ARG. And that's the kind of problem I like. So let's go on to the next one. Question number 13. The following question has 2 parts. First, answer part A, then answer part B. Part A. Students investigate the effects of a single base mutation that occurred in a piece of DNA. The diagram shows the first 15 bases of the DNA template that they are investigating. A thymine nucleotide is mistakenly added between the adenine and the cytosine at the location marked in the arrow. So let's look here. Here's our DNA. And you can see where the base was added between the A and the C. Okay, and that would be a T that's got added. So let's go here. So which messenger RNA sequence is produced from the mutated DNA template? Well, if we start off with TAC, trees to apples, apples under garage, cars, it would be AUG. So that's D. That's got to be our answer. But because there's nothing else that begins with AUG. But let's follow what's happening here. So AGC would be UCG, which would be here. Now let's see what happened when we had the mutation occur. So this would be U for apples, and then UAG. UAG is correct. So let's look over here. UAG. Oh, that's a stop codon. So the process is going to stop then. So this is our answer. Now let's go to part B. Select all the effects of the mutation on the protein produced. Well, since there's a stop codon, the protein is going to be shorter, and the protein, because the amino acid sequence is going to be different, will function differently. The protein will gain an additional active site? No, it's been shortened. Nothing's going to be additional. The protein will have more complex structures? No, it's not going to be any extra things involved with it because it's gotten shortened. The protein will be the same length, but will have different amino acid sequences. No, it won't be the same length because of the stop codon. So our answer for this, choosing all the correct answers, would be the first two. Okay, thank you. Question number 15. A protein is built from several polypeptides, each with different three-dimensional folding pattern. What causes the polypeptides to have different folding patterns? Well, first of all, to understand this question, you got to know that word polypeptide. So poly means many. When we go from DNA to RNA to protein, and DNA gives messenger RNA a code, that code is for an amino acid. And when these amino acids are put together at the ribosome, that's called a polypeptide chain. So we know what's happening here now. So what causes the polypeptides to have different folding patterns? A, the polypeptides are assembled by different ribosomes. The polypeptides are assembled in different parts of the cell. C, the polypeptides are composed of different amino acid sequence. Or D, the polypeptides are controlled by different gene expressions. Well, the answer is the polypeptides are composed of different amino acids. So if your DNA gives messenger RNA a different code, you're going to get different codes for the amino acids. They'll be in different sequences. Okay. You can eliminate A because the polypeptides are assembled by different ribosomes. Ribosomes have nothing to do with it. The polypeptides are assembled in different parts of the cell. That doesn't really mean anything. And D, the polypeptides are controlled by different gene expression processes. So guys, the polypeptides are composed of different amino acid sequences, and that makes for a different protein. Question number 19. Researchers studying hummingbirds in the Andes Mountain of Ecuador made several observations. There are many species of hummingbirds. The species are not separated by any physical barriers. Unique hummingbird species are found at the different elevation in the Andes. Which hypothesis is based on these observations? A, conditions at each level favor different hummingbird traits. B, all hummingbird species are able to interbreed at every elevation. Species don't interbreed with each other. C, elevation changes causes hummingbirds to develop new methods of transportation. D, individual hummingbirds choose food sources based on their preference for specific plants. The correct answer is A. Conditions at each elevation favor different hummingbird traits. So whatever conditions exist at the different levels, because of evolution, the hummingbirds will, of course, evolve to fit into that environment. So the correct answer is conditions at each elevation favor different hummingbirds. Osprey ecosystem. Osprey are predatory birds that typically nest in open high places. Scientists monitor the open nest of a native osprey population in an estuary habitat affected by human activities. The scientists observed a natural occurring feeding relationship in the osprey ecosystem as shown in the table. So we have the ecosystem feeding relationship. These menhaden small fish, their food source is, of course, plants. The striped bass is the menhaden, the herring. The osprey is also the menhaden and the striped bass, and the raccoon is the young osprey, eggs, frogs, and nuts. The scientists noted the following events affecting the ecosystem. A. The striped bass harvest increased with an increase in local commercial fisheries. B. Due to overfishing, restrictions on striped bass harvest were enforced to reduce effects on the population. C. A conservation project restoring eagle populations from a similar ecosystem has introduced eagles into the osprey ecosystem. And D. Eagles fill the same niche as the osprey. So it looks to me that our osprey is going to have competition from the eagle. The graph shows how these events within the ecosystem have affected the osprey population over time. So we have time going at the bottom, we have the number of ospreys at the top. And as you can see in time, the first segment of time, the osprey populations are growing. The second, it's going up and down. But in the third, when I assume the eagles were introduced, the osprey population started to go down. So let's look at our question. Which statement describes a possible effect of the introduction of the eagle into the osprey ecosystem? A. Osprey mating calls will change in tone and frequency. B. Osprey migrations will become shorter and less frequent. C. Osprey will produce fewer eggs in each breeding season. Or D. Osprey will become more selective for fish during hunts. The correct answer is C. Osprey will produce fewer eggs in each breeding season. Why? Because they're going to have competition from the eagles to even exist. So, osprey mating calls in A really wouldn't be wouldn't be involved with this question. Osprey migration will become shorter and less frequent. We have no proof of that. And D, osprey would become more selective for their food. No, they would become less selective because they're fighting to survive. So the correct answer is osprey will produce fewer eggs in each breeding season. Okay, thank you.